Good afternoon, ladies and gentlemen. We are now going to pass to a session that is dedicated to the successful use of Instruct-ERIC. That is the successful use of Instruct facilities by the Portuguese community. Because after all, that's the mission of an infrastructure. Infrastructure provides solutions for problems from the users. So I'd like to call the first speaker. That will be Tiago Santos. Targeting of building partnership with comes from the Universidade Beira de Minar. Cellular. A warm welcome to him and the stage is yours.
Thank you for the nice presentation. Hello everyone, good afternoon. My name is Tiago Santos and I am a PhD student at the University of Beira Interior, located in the center of Portugal, in the small city of Covilhã. First of all, I would like to acknowledge the organizers, and especially Professor Maria Armenia, for the kind invitation to present our work and to share our experience as you. Exactly. To share our experience as users of the Instruct-ERIC platform. First, we thought to use the platform as a service to produce a protein. However, with the development of the work, we have established a fruitful collaboration with the Cell-Free platform located in the Institute of Structural Biology in Grenoble. Today I will talk a little bit about some of the work developed, and my presentation is entitled Targeting of Nucleology with a Complex Partnership. My presentation is divided in the following way. In the first stage, I will talk a little bit about Plexus, how its structure, its function, their binding and stabilization by small molecules that we usually call G-Plex ligands, and also their binding to nucleoli, a protein that is overexpressed in the surface of cancer cells. In the second stage, I will share the results, the experimental results, that prove the formation of RNA G-Plex in the sequence of pre-microRNA 149, as well as its stabilization by ligands and its binding to nucleoli. Finally, I will present the main findings and conclusions of our study. So, first, it's important to describe RNA G-Plexus. The human transcriptome contains many sequences that are rich in proteins that can fall into non-canonical three-dimensional structures that we call G-Plexus. G-Plexus are characterized by the presence of two or more stacks of formulins organized in a coplanar manner. Each set of formulins forms a building block that we usually call a G-tetrad. G-Plexus are only formed in the presence of metal ions, usually potassium or sodium. Regarding their functions, RNA G-Plexus are involved in many cellular processes, such as telomere homeostasis, stress response, RNA translocation, interaction with RNA binding proteins. RNA G-Plexus also play a fundamental role in microRNA biogenesis, an important pathway to regulate gene expression. G-Plexus in this pathway could inhibit microRNA biogenesis by reducing the levels of mature microRNAs. G-Plexus could be found in primary, in precursor, and in mature microRNAs. We have studied the G-Plex motifs of three pre-microRNAs, precursor microRNAs, namely LED7E, 92B, and 149. However, in this study, we only focus on pre-microRNA 149 because its major form is dysregulated in several cancers, and its sequence and the sequence of its precursor have the ability to adopt a G-complex structure. The stabilization of the G-complexes in microRNA biogenesis could be achieved with small molecules, with ligands, and in the last years, several small molecules have been designed and developed. For instance, pyridoxastatine and FNDC3 are found to be good stabilizers of the G-complex structures, while porphyrin derivative is a molecule that can disrupt G-complexes. All of these small molecules share structural features like an aromatic core that could establish stacking interactions with the G-complex, with the outer G-tetrads, could stack on the top and the bottom, and also an aliphatic side chain that could establish interactions with loops and grooves. In this work, we have only used two accreting derivatives, C8 and C8NH2, that were previously synthesized by a collaboration that we have with Yeast, and also have been tested in oncogene promoters such as CEMIC to stabilize the G-complexes in oncogene promoters such as CEMIC and KRAS. Next, beside ligands, also proteins have been found to bind to G-complexes, to RNA G-complexes. Nucleolin is one of those proteins and are involved in many biological processes, as well as pathologies like cancer, neurodegenerative diseases, and viral infections. However, the structure of nucleolin is not straightforward, it's very complex, and it contains a long disorder region in the N-terminal domain and a small one in the C-terminal domain. The protein also undergoes post-translational modifications such as methylations, glycosylations, and phosphorylation, which in addition to the long disorder regions difficult the expression of the protein. Because of that, until now, only the solution structure of domains 1 and 2 of the protein has been studied until now. Indeed, this region is the most important for the G-complex recognition and binding. Taking these concepts into account, we developed a proposal to apply to an Instruct-ERIC access call, where we propose that our partners from Institute of Structural Biology in Grenoble express nucleolin in a cell-free system, and then we characterize the binding of the protein to the G-complex and to the G-complex C8 complex. However, this collaboration was recently extended to other projects, to other microRNAs that have been identified as G-complex forming molecules. First, I will show you the results of the cell-free production of nucleolin labeled with carbon-13 and nitrogen-15. Our partners from the cell-free platform tried to produce a full-length nucleolin. However, the in vitro expression was not achieved with success because of the disorder region in the N-terminal domain. Taking this into account, they tried to express domains 1 and 2. They are successful, which were these two domains were previously described as RNA binding domains, and we continue with these two domains. The purification of the protein was performed by us by using an IMAX strategy, and in the final we have a solution of protein with high purity. Now I will share the results of the formation of the G-complex in the presence of potassium. As a first approach To characterize the formation of the G-plex, we take advantage of NMR spectroscopy, which gives important information about the G-plex immuno pattern. The signals of the G-plex only appear between 10 and 12 ppms, while the other secondary structures of nucleic acids appear between 12 and 40 ppms. As seen in the slide, the G-plex concentration at the G-plex concentration of 100 micromolar, the presence of 100 micromolar of KCl and at a temperature of 20 degrees, the structure is well folded, as we can see by the sharp and well resolved edges. We also changed the concentration and the temperature, and we found that at concentrations of oligonucleotides higher than 100 micromolar, or at concentrations of KCl superior to 100 micromolar, the structure aggregates. Okay. Next, we assessed the stabilization of the G-plex ligands C8 and C8NH2. For this reason, we used CD melting experiments, which can show the melting temperature in the absence of the G-plex, in the absence of the ligands, and in presence of the ligands. And as we can see, the ligand C8 demonstrated a remarkable ability to stabilize the G-plex structure. Taking this into account, we only proceed our studies with C8 and not with the C8 and H2. Next, we conducted our studies and performed STD NMR experiments. The method consists on the transference of the magnetization from the macromolecule, in this case the G-plex, from the ligand, in this case the C8, which permits to unveil which atoms of the ligand are in close contact with the G-plex. As we can see in the slide, the aromatic core of C8 and the iodine ring are the parts of the ligand that are in close contact with our G-plex, which is expected since the ligand seems to stack over the G-plex. Then we performed 1D NMR titrations. We titrated the solution of G-plex with the ligand, and we found that a concentration at a ratio of 1 to 0.75 of G-plex ligand, the complex is totally formed. In this way, we have conducted molecular dynamics studies to predict the binding mode of the ligand on the G-plex, and we found that the ligand stacks over the top G-tetrad, establishing mainly stacking interactions. Then we evaluated the binding affinity and thermodynamic parameters of G-plex and the G-plex C8 and the binding with nucleolin. To this purpose, we have performed fluorescence spectroscopy to follow the fluorescence intensity of the G-plex labeled while we titrated nucleolin. And the results reveal that in the presence of C8, the binding affinity is significantly better. We also evaluated the thermodynamic parameters, and the KD was in the micromolar range. A stoichiometry of 1 to 1 was achieved, and an enthalpy value of less than 34,800 kilocal per mole was obtained. Altogether, these two techniques proven that the G-plex bind to nucleolin with high affinity. Next, we evaluated the binding of the G-plex to the domains 1 and 2 of nucleolin by 2D NMR and molecular modeling. And to this purpose, we have used the domains 1 and 2 of the protein that was produced by our partners from an Instruct-ERIC platform. Again, the results show the strong interaction of the G-quadruplex with the protein, as seen by the loss of intensity in the signals that are enclosed in the red. The most affected, interestingly, the most affected signals are those that are pointed to the linker between domains 1 and 2, or even in the linker. And this is because the residues of the linker is positively charged and possibly interact with the backbone of G-quadruplex that have a negative charge. By the end, we evaluated the cellular uptake and the cytotoxic profile of the complex G-quadruplex C8 in prostate cancer cells and in healthy cells. To this purpose, we have used confocal microscopy. We observed a significant cellular uptake of the complex in prostate cancer cells, but not in the healthy cells. Taking this into account, we proceed the cytotoxicity assays, the MTT assay, and we show that a remarkable synergistic cytotoxic effect was observed in cancer cells in the presence of the complex, but not in the healthy cells. As conclusion remarks, we would like to point out that we already published one scientific article with our partners from the Cell-Free platform, and 2 new publications are scheduled. We also would like to conclude that NMR and computational studies predicted the binding mode of acrylamide derivatives and nucleolin to the G-quadruplex. C8 demonstrated a remarkable ability to stabilize the G-quadruplex found in microRNA 149. Structural studies revealed the binding site of the G-quadruplex on the domains 1 and 2 of nucleolin, and the confocal microscopy and the viability assays revealed a high selectivity of the complex to cancer cells. By the end, I would like to acknowledge my supervisors, Professor Eurico, and our collaborators from the Cell-Free platform, as well as the funding provided by Instruct-ERIC Access Platform and FCT. Thank you.
Thank you very much. So at the end of the session, we will have a discussion of this presentation. Thank you. Thank you. Now I'd like to call that will talk to us about exploring a new drug target against Mycobacterium tuberculosis. So it comes from the Latin scene and it resides at the University of
Hello, my name is Hugo. I'm going to talk about the project in fact, 3 projects that were extremely useful for someone like me that's not a structural biologist. I've been working for some years with a protein from tuberculosis. As you know, tuberculosis it's still a major disease in the world. 2 to 3 billion people are infected worldwide, and about 50% to 50% of these people become active. Portugal has the highest incidence of TB in Europe. The treatment it's a vaccine. In the past, everyone in Portugal has a vaccine. Currently, only a few regions of Portugal have that take it, and a combination therapy of several antibiotics that can extend for 6 months. The problem, as you all know, is that currently there's a lot of drug resistance, antibiotic resistance, and this creates a need for antibiotics and for new strategies for killing teeth. I've been working on this drug target for some time. for some time. This drug target is essential protein in tuberculosis. It's a protease. In fact, it's quite hip now. It's also important for cancer, for example. But it was first discovered in Goldberg lab in the US in the last century, and it's basically a protease. It's a serine protease. It has a classical mechanism for a serine protease, but it works different in the sense that it's an enclosed protease. It's basically a cylinder, and with the active sites inside. And this makes sense because the idea is to prevent uncontrolled proteolysis. So you want to have protease that's regulated, and this regulation occurs through activators. These activators are a group of proteins. Here I call them chaperones. Some people call them unfolded. It depends. And these proteins, what they do, they have several of them. They work by recognizing the substrates, unfolding and translocate them inside of the clip to the group. So it's a very ingenious way of controlling. So it prevents the fact that the active sites are inside clip B, prevents the protease to be active per se, and the degradation only occurs when these activators, these chaperones, bind to clip B and they feed the substrate inside. The bottom you see a nice cartoon. This shows basically, it's very simplified obviously. This requires cycles in ATP. So basically these activators, clip X, clip A, clip C, they use ATP to unfold substrates and translocate them inside the protease where the degradation occurs. The protein I've been working for some years. In the title I put new, in fact, I've been working for more than 6 years on this target. It's called CLIP-C1. CLIP-C1 has the same, works exactly like I mentioned before. It binds to clip P and inserts protein substrates inside, and these proteins are degraded. What's interesting about this CLIP-C1 inhibitor is the fact that we have so far 4 natural product antibiotics that act on this protein. These obviously essential protein TB. This is very obviously it's very tempting for drug development, protein like that. First of all, you have already molecules that bind to it, and moreover they're quite potent. So this is it's not really a model, obviously. This is a model of CLIP-C. It's an hexamer, so it's called 16 units. And it has this kind of shape. What I call here NTD is the anterior domain. This is D1 domain, D2 domain. What is really important for the rest of the talk is that this NTD doesn't have enzymatic activity, but this D1 and D2 are the ATPase sites. So this is where the ATP hydrolysis occurs. This is the sites that you have that generate force. That's going to help the unfolding of substrates. You can see that from this top view, the substrates basically they enter to this pore and they are pulled like a rope. And this force is responsible for the unfolding and for feeding, for injecting this peptide inside of the protease. Then the protease just cuts it. So far, obviously, the drug target, you're going to have structures, you're going to understand how these things work. There's a lot of work here in the domain. The NTD domain is very easy to express, it's very easy to purify, it's very easy to crystallize, it's fantastic for structural biology. And in fact, most of the antibiotics, in fact all of them, most, bind to this NTD domain. So this is a figure from one of our papers. And basically I put only 2 of the antibiotics. There's 2 others, they all bind to the NTD domain. The problem, of course, you understand how these drugs work. It's a bit difficult because the NTD domain doesn't have enzymatic activity. So we would like to understand how these antibiotics work in a sense that they bind to the NTD, how they affect the activity of this gene. The problem is, of course, we tried to crystallize the full-length protein. It didn't work. We tried several times. It's a tricky protein to work with, not very soluble, and moreover, it never crystallized. Of course, with products, you never know if you use all the conditions possible. You can stay forever trying to crystallize a protein. We did some SAPs in granola, was around for a long time, and we figured out to discover at the time that in fact this protein that was supposed to be an hexamer didn't elute as to run as an hexamer, right? Like it was much bigger. Our results came at the same time. Our results from the Mycobacterium group in Germany, when they identified a resting state for this kind of proteins. So in fact, this protein doesn't really exist in the cell as an hexamer, but it's distributed between a resting state that has this kind of shape, not a bit more flat and difficult to describe. And we thought at the time this would be the reason why we like this protein, that the protein is heterogeneous. In fact, this is also results from the Mycobacterium group. They did some cryo, not very low resolution, at 10 angstroms. And what was curious, they proposed in this paper, it was in Arius, in this work that was doing in Arius, that this middle domain that stays between D1 and D2 was responsible for the formation of this resting state. And they proposed some restings that were important for the formation of this resting state. Our hypothesis was very simple. Maybe by mutating these residues in TB, using our target from TB, we could just shift the protein to the hexamer state, and we were able to get some structural information on our target. I put this is basically a joke, but this is really what I do, because I'm not really a structural biologist, I'm a biochemist. I don't do structural determination, I basically do protein purification and enzymatic characterization. So what I did in this project, this is a short story, I introduced some mutations, I purified it, I characterized it biochemically, I'm going to explain after, and then I used the infrastructures from Instruct-ERIC to get a structure. So basically our idea is that doing certain these mutations, we could change the equilibrium from this resting state to this hexamer, to the active hexamer, and in fact it looked like that on gel filtration. So this is basically it's a statistical chromatography with a mutant, and basically what you see, we see a shift in the size of the main species to something more agreeable with what to expect from the hexamer state. I put here also these welcome motifs, because this is the ones that basically, since you're gonna try to stabilize the hexamer and the hexamer is continuously agilizing ATP, what we do, and this is quite common in the literature, is to mutate these motifs and basically freeze the molecule in a state that it cannot utilize ATP, so ATP is gone. So I did some enzymatic characterization. I did this basically because I wanted to show that the protein was still active, because I know if we ever try to publish this, the first thing is I mutated the stuff, it's good for nothing. So basically, what I did is try to prove that in fact this mutant was still active, was still capable of basically neutralizing ATP, of catalyzing protein degradation, and also, very important, able to respond to antibiotics. So this is an ATP, as I say, this is quite classical in literature. Basically what you measure is ATP hydrolysis. In this case I used the fluorescence, you can also use absorbance for NADH, it's similar. What I want to show you here is in fact these mutants are active, so they are able to hydrolyze ATP. And in fact they are still responsive to the antibiotics. So I tested two of them, the ones I had, so it's Cytheratic and Mycobacterium. And it shows that this mutant, despite being an modification, obviously is always a modification mutant, was still important was still going to give us some information how these drugs would work, and they didn't abolish the function of the enzyme. Another thing that's really important is to test the full thing, to test if this CYP1 was also able to bind to CYP, to the protease, was also able to catalyze protein degradation. And this is more difficult to explain, but I'll try to be clear as possible. So basically what we do here, we use model substrates. One of these model substrates is casein. Casein doesn't have a defined structure. So it can be considered IDP. And it's used in these assays because it can be modified with fluorescein. So what we do here, you have a protein labeled with a fluorescent tag. As the protein is degraded, you have an increase in fluorescence. So what happens basically here, you mix everything, you make your chaperone, your amphylase or chaperone, as you want to call it, and you mix the protease, and then as the protein is degraded, you have an increase in fluorescence. What you show here is that yes, that this mutant was still active, still working fine. Another type of assay is just that you all know GFP, I guess. GFP is an intrinsically fluorescent protein, very, very stable in contrast to its casein. And basically the idea here is as GFP is recognized by CLIP-C1, is unfolded and translocated inside of CLIP-B, is separated into pieces, and of course you have a net decrease in fluorescence. And you also see, you still see the response of the antibiotics. That's our goal, is to understand how the antibiotics work, so that was extremely good. So, how this worked? This was done during the COVID time, and I was really surprised how easy it was. So I'm making a great advertising for this iNext and Instruct system, because I applied to CSIC, and with the help of Teresa, it was during the COVID time, so I didn't go there. We shipped samples. They even paid the shipping. It's true. It was not false advertising. They paid the shipping. They received this. They made all the samples. They made the grids, and they also via the Instruct, they also did the processing. This in a very fast way, in a very easy way for me, because I'm really not an expert in this. So don't not ask me questions about technicalities, because I'm not able to reply to them. What I can tell to you is that these people were really efficient and really easy to work with, and I definitely recommend to try it out. I think I had a big part of luck in the way things turned out, but I think the way the things evolve so fast and easy, I think definitely is worth trying. So, going back to what's important, they also did negative stain in Spain, so you can see these examiners there. The grids was done by Teresa Bueno, and then Marcos did the processing, and Marcos was extremely efficient. Basically, we had a talk by Skype. He said to me, Hugo, we have these 2D averages. Do you think it looks like your protein? Of course, this is models with the literature to see what this thing should look like. And in fact, we could kind of, this is the original pictures I sent to Marcos. Yeah, I think it could be. And in fact, you can see that yes, the 2D averages look very much like the models we're expecting. And then Marcos did the processing. Since this is the original spread, the process is done by Marcos, then a colleague of mine in Belgium, Jan, took over and to do some extra processing. And in fact, we were able, by this fast procedure, we were able to get the structure. I would like to tell us just a single point. We cannot get some parts of the models we can see. This is just a joke, because we're AlphaFold. I really love AlphaFold. I'm a really big fan. I'm not to be sad, in fact. I'm not trying to I'm just saying, this is what AlphaFold predicts for our protein. This is what we see with our structure that I'm going to show after. Some parts are missing. Sadly, the MTT, that's where the antibiotics bind. Also, the middle domain, we don't see it. All the rest we see, and that's what we get. What we got, and this we got 3 structures in very good resolution. We got the apt structure, the cyclomer structure, and the combination structure. I will talk about this one more later. This is you see the D1, D2, you see the nucleotides bound, you see 6 nucleotides in the D1 ring, 4 in the D2, and spectacularly you also see full peptide. This was unexpected, but it was already reported in literature for proteins of the same family. You also see the tyrosines of the binding loops surrounding these peptides. So this is basically a snapshot of our machine working. Okay? Sadly, we don't see the entities. One thing I would like to talk very briefly, because it's still ongoing, is some of the particles in ecomycin. It's one of the antibiotics. We do see the entities, at least some of the particles. There's electronic density here, so we still work in place. So we think for the ecomycin, for the antibiotic, we may have a more complete picture of our machine. And this is my speculation, or you can call it a good speculation. I think, for example, it's going to explain how the mechanism of action of ecomycin, because ecomycin is a big molecule. It may mimic a substrate by binding to the entities and put them in this position. But of course, this is completely. We need more data to support this speculation. The team. I would like to thank Instruct, iNext, of course, and I don't know how to say it. I have to recommend it, obviously. Also, I would like to thank Instruct for they sponsored me a pilot project some years ago. while I was in Grenoble. Catarina is also working at USS in Porto. She also got a pilot instruct to go to India, Hamburg, also to the cryo. Teresa and Marcos é o espetáculo. Raquel Soares is my boss, and Jan is helping the processing. So, very much for that.
So now I'd like to call Isabel Schwab from CDMN, that will talk to us about the three-dimensional structure prediction on molecular topic of the study of carboxylic.
So thank you very much for the invitation to be here. I'm a bit of an outlier because I haven't used the Instruct facilities yet, so I hope that in the next meeting I will have some data resulting from this very interesting platform to present you. So why are we here? Because we are really interested in the structure of membrane proteins, in particular carboxylate permeases. So we have been studying carboxylic acid transporters in our group for several years now, trying to understand structure-function relationships, but also how they affect physiology and how they can be used in biotechnology. There are several types of mechanisms to transport carboxylic acids in yeast. The non-dissociated form can can cross the plasma membrane without the aid of a protein, but when we have the charged molecule, we have to have a mediated transport system. In our group, we are focused on the study of permeases. So our model system is Saccharomyces cerevisiae, and when we have yeast cells grown on media containing acetic acid or lactic acid, we have the expression of these transporter systems. And throughout the years, we have characterized several of these systems. One is the gene 1 family that was initially characterized as being encoded. It's a lactate proteins importer that has been characterized by Margarida Casal in 1999, just to give you a perspective of how things evolved in our group. And then throughout the years, we characterized several other yeast transporters. Last year, we focused on a specific yeast that has 6 copies of this gene, and so we are interested in understanding how they have different specificities, different affinities, and how they evolved as well. So back in the beginning, we searched for gene 1 conserved domains, and we found this really interesting one in the 7th transmembrane segment. We decided to do Cytheratic Mutagenesis and explore why we had homologous proteins with different transporter specificities. So here we have the wild type strain that grows on lactic acid very well, and when we delete gene 1 gene, it loses the ability to grow using this carbon source. So when we do specific mutants, we got very interesting phenotypes, and we were in fact able to recover phenotypes that recognize another carbon sources, such as succinic acid, which is the carboxylic acid, a much bigger molecule. So at the time we were still in 2007, so we were learning how to predict structures because we didn't have that much crystallite structures of permeases, and we understood that this was a powerful tool, because after getting the results with the mutants, we saw that the predicted location of these amino acids was right facing the pore of the protein. We then did some substrate docking, and then we unraveled the substrate translocation pathway predicted that corresponded exactly to the amino acids that we had uncovered with our strategies. And then we uncovered some new residues that also gave us some very interesting results in terms of affecting the specificity and affinity of the protein. So at that point, we got a set of mutants, and we could uncover some residues that were really affecting the overall function of the protein. We then focused on the other carboxylic acid transporter, which was encoded by ADY2 in Saccharomyces cerevisiae. This is just a plot where you see that when you remove this gene from Saccharomyces cerevisiae, it's not able to have a mediated transport system for acetate, while in the wild-type gene you have this mediated system. And after this gene was characterized in cerevisiae, we were able to characterize several homologs, namely in E. coli and also in fungi, in Aspergillus, and in Archaea as well. We had a collaboration ongoing. We started to uncover all the homologs existing in all the reference genomes available at NCBI. We uncovered 800 homologs, and it's quite conserved throughout the fungi, so it plays a very important role in fungi, and it's also present in several prokaryotes. By doing it as well an identification of the conserved residues, we identified a conserved domain located in the first transmembrane segment, and when we do mutations in this conserved domain, again we have interesting phenotypes. You can see here that while the wild type grows very well on media containing acetic acid as sole carbon and energy source, when we do mutations in these residues, we decreased considerably the transport, and also this is the uptake of acetic acid. It's also decreased while not affecting the protein localization. So we saw that this was an important region of the protein. So at that time, Professor Margarida had a collaboration with Professor Margarida Archer, and they tried to, they were successfully producing the bacterial homolog of ATO1, but then there was this group from China that got ahead of us and got the crystallized structure of this permease that they call a channel as well. So they got the crystal structure for the SAT-B, which is the homolog from Cytheratic Mutagenesis, and then another group from E. coli, and it has a predicted structure of an hexamer, but the functional transporter is the monomer with 4 predicted binding sites. So what we did was to model our ATO1 from cerevisiae, and we also got the same 4 predicted binding sites, and at the time what we decided to do was to explore this fly site, which is located in the middle of the protein, and it's the narrowest constriction site present in this permease. And we tried to see if we could somehow modulate the activity of the protein by doing some alterations in its overall structure. So what we got was, this is just an example, and by manipulating or mutating the leucine residue of the fly motif, we're able to, you can see here that this is the wild type that grows a bit on acetate and does not grow on lactate or succinate. which are bigger molecules of carboxylic acids, and when we decrease, or when we mutate to alanine, we start having growth on these substrates. So meaning that the transporter is able to transport these substrates. So you can see here as well, this is the transport of acetic acid. When we have the wild type protein and mutated is the same for acetic acid. But then when we have for lactic acid, it transports a bit and starts to transport 3 times more than the wild type, and then for succinate, it's also a big increase. So we decided to play a bit with this amino acid in particular, and by doing mutations to other residues, we got a different set of phenotypes that we decided to explore a bit more. So we predicted the pore of the different mutants, and if you look here, this is the wild type. You can see here in red, it's the nearest constriction site that is generated with the fly residues, and then when you shift this to an alanine and a glycine, valine, serine, etc., you have an increase of the constriction site, and then you have here a complete blockage of this spore, and it's also presented here. And what we saw was that here it's the wild type that transports acetic acid, and when we start to decrease this residue in particular to valine, we start to have a much higher capacity to transport lactic acid, and then again when we decrease it to smaller residues, we have the transport of succinic acid, which is a bigger molecule. And then here we have a complete decrease. We don't see any activity when the residue is much bigger, although the protein localization was completely correct, so we did not affect protein expression. And so this for us was very interesting from a biotechnological point of view, because we are interested as well in uncovering transporters with new capacities, new specificities to be expressed in microbial cell factories. So what I want to say is, as a final remark, that this is a very, using the 3D structure prediction and the substrate docking, we were very able to uncover structural features of our transporter proteins. Nevertheless, and being here and hearing all the possibilities of the Instruct and all the facilities that are available, we want to take the next step and to unravel the overall structure of very interesting targets that we have uncovered in our lab recently and don't have the same robust models that we can work with. So finally, I just want to acknowledge the research group and some of the PhD students. 2 of them are here with us that did a lot of this work, and Professor Margarida Casal, who started this work so many years ago, and it's been very interesting to see how our knowledge in transporters has evolved throughout the years. So thank you very much.
Thank you very much, Isabel. So now I'm calling Cytheratic Mutagenesis that will be talking to us about targeting cell wall synthesis in Mycobacterium. The immunoforensic transferases as target for tuberculosis drug.
Thank you very much for the organization of this event, for the invitation. I will talk about targeting cell wall synthesis in Mycobacteria as target for tuberculosis drug development. So, as you already talked about, the tuberculosis is an infectious disease caused by Mycobacterium tuberculosis. It's still one of the leading causes of death worldwide, And right now, it's the second leading cause of death caused by a single infectious agent. Only COVID-19 ranks above TB. I will address the burden that COVID-19 had on the tuberculosis worldwide. So for the first time since 2005, there was an increase in the death toll of tuberculosis. 1.3 million people died. And there is also a decrease in reported cases of TB. The estimation is 10 million new cases appear each year, and only 5.3 million cases were reported in 2021. So the cell wall synthesis of Mycobacteria is a unique structure and very complex. It has an increased thickness and hydrophobicity compared to other gram-positive bacteria, and it has lower permeability to drugs. So this makes, and it is an essential feature of the bacteria. So therefore, it is a suited drug target against tuberculosis. There are already drugs against TB that act on several layers of the cell wall. So isoniazid acts on the glycolic acid synthesis, and ethambutol acts on the arabinogalactan synthesis that I will focus in more detail. So the proteins that we study are arabinofuranozole transferases, so they are membrane proteins, and they transfer arabinose residues to the growing chain of arabinose. So most of these proteins are essential. Some are not, but most of them are essential to Mycobacteria. And we can classify these membrane proteins as far as the inhibition towards drugs. So some are inhibited by ethambutol. So EMBs are susceptible to the first-line drug ethambutol, but AFT, A, B, C, and D are not inhibited by any known drug, so they are novel targets against tuberculosis. So, in essence, the 3D structure of these proteins will be a powerful tool to design new drugs against tuberculosis. So, the first one we were involved with is AFTD. So we determined the structure of AFTD from Mycobacterium abscessus. AFTD is the largest predicted RAT encoded in the genome. It has 150 kilodalton size, and we expressed in E. coli, purified in detergent, and incorporated into nanodisk. And we used single particle cryo-EM to get the structure, because the yields of these proteins are very poor. So this work was in collaboration with Columbia University. We solved the structure with 2.9 extra resolution in a Titan Krios. So the overall architecture is like this. On the periplasmic side, we have carbohydrate binding motifs to bind to the arabinose chain. On the interface between the periplasmic domain and the transmembrane domain sits the catalytic site. We have the several transmembrane helices. This domain is important because the donor of arabinose is lipidic, so it comes from the transmembrane domain. And to our surprise, on our structure we had a bound acyl carrier protein from the host organism. In this case it was E. coli, ACP, and we were very puzzled about that, and I will talk about it on the next slide. As far as the catalytic site, even though this was an apo structure without any substrate bound, by structure comparison and structure conservation with other glycosyl transferases, we identified several amino acids involved in the catalysis. And in fact, when we mutated those residues, the mycobacteria started it impaired the growth of mycobacteria. As far as the bound acyl carrier protein, we found an extra density that it is the phosphopentatine arm, which was buried into these 4 helix of the transmembrane domain of AFTD. We couldn't find any cargo, any density that we could associate to the ACP carrying something. And we wanted to study this interaction. So we also designed a few mutants to try to abolish this interaction. And they also affect mycobacterial cell growth. So it is somehow important to the function of the protein. So we were puzzled with that. So one particular mutant was very effective and affected the cell growth. So we also determined the structure by cryo-EM. And we weren't successful in abolishing the interaction. So if you can see the wild type, you can see the ACP density here very well defined, and the mutants, even though it's not well defined, it's fuzzy. So the ACP is still there, but it's more flexible. So therefore, we couldn't model the protein to the density. Not only that, a few transmembrane helices get disordered with this mutant, and to our surprise, one loop that we couldn't model in the wild type structure becomes an ordered helix and blocks the catalytic pocket. So this is the main difference between wild type and mutant. And so we think that ACP may be regulating AFTD activity because when we abolish or impair the interaction between ACP and AFTD, there is conformational change and a blockage of the catalytic site. Right now I'm working on AFTA from Mycobacterium tumbellini, and this is the most challenging project so far. So AFTA is the arabinophrenosyl transferase that transferred the first residue to the arabinine chain. So it is one very interesting drug target in that sense. It is way smaller, so it is a 68 kilodalton protein. We purify in the same manner. We purify detergent and incorporate it into nanodisk. However, the problem with this particular protein, despite the low yield, is common to every single membrane protein that I work with. We had problems with concentrating this protein, so it precipitated a lot. So we couldn't reach the concentrations suited for crystallization, and the overall size makes it too small for cryo-EM. So through the Instruct Center in Brussels, we went there to discover nanobodies against this protein, and the strategy for single particle cryo-EM is to increase the size of the particle using first nanobodies and then upgrade the nanobody to megabody, and hopefully get a protein complex, and we solve the complex altogether with single particle cryo-EM. So the megabody that I'm working with is a 100 kilodalton soluble protein, so it bulks up the particle size in a significant manner. and it's highly specific with AFTA. And we purified the complex using differential density gradient ultracentrifugation, in this particular case a sucrose gradient, where we incubate AFTA here with the megabody, and this is the scaffold protein for the nanodiscs. So we load on top of the gradient, and the gradient is able to better separate the empty nanodiscs from the nanodiscs that contain my protein AFTA. But also we can more or less see and select the better ratio between the three components of the complex. So we can select the best fraction to go to the microscope. So in the end, go back. So in the end, this is the last, the final sample. So very pure, more or less the intensity of the megabody and the FDA, it's more or less even. And we went to the Instruct Center of Finland to collect data on this sample in the Thales Arctica, and so far I was able to identify two populations in my sample. Unfortunately, the most prevalent population is the megabody alone, and the second population, it's about 1% of the total particles that we got in this data collection. But as you can see, there is some extra density, and it's bigger than the second population. And this, we assume that it is the AFTA megabody complex. So this is the ab initio model of that complex. I'm actually thinking of making it some sort of a vase, porcelain vase, because it's quite beautiful, Art Deco for my living room. But other than that, it's really a low resolution model. However, we could fit the megabody PDB that already is available. And as you can see, it fits well in the boat, like the boat-shaped density. Yeah, people can see it, no need. And they will believe my word, because I'm looking at this thing a lot lately. So, as future work for this AFTA, we would like to improve the sample quality, maybe increase the ratio of AFTA to try to get more population that does have AFTA in there, and collect more data, like more Krabian data. For that, we rely on Instruct centers for that. Last but not least, we were also involved in the determination of ENBB structure for Mycobacterium smegmatis. However, we got more or less scooped because another group actually published all three ENBs, ENBA, ENBB, and ENBC from tuberculosis. That is the holy grail of what we want to achieve. Nonetheless, it was really good for us to see that our first, the first report of the ACP interaction for AFTD, they found also a ACP bound to all those structures. So in this sense, we were very happy to see it. But right now, with the published structure, we can do in-depth study regarding ENBB, which has a lot of mutations that are clinically relevant to TB patients that do have drug resistance against some drugs. And we right now are using the template, the Mycobacterium tuberculosis structure with the drug-bound ethambutol on the catalytic site, and we're doing redocking with the drug. We're trying drug repurposing and molecular dynamic simulation to see and get more insight on the effect of these mutations. So, starting with the redocking, the results were very promising because in green you can see the experimental data density of the ethambutol drug on the structure, and as you can see, the hits that we got more or less are on top of that location of the experimental data for ethambutol, but in terms of the analysis, we see that the docking results for the wild type have less clusters. With clusters, I mean less possible position for the drug. While when we do in silico mutagenesis to valine, the number of possible positions increase. So, and also the binding energy decreases, but we take it with a grain of salt. But the clusters, the increase of clusters is already a prediction that it may be more mobile, the compound within the cavity. And we do see that with the molecular dynamics. So right now you're seeing the wild type, and this is the ethambutol drug. And on this side, you see that the drug slowly starts to leave the cavity, and eventually it does leave the cavity. And this is very interesting, and this is not very well described in the literature. So right now we are trying to do this approach with a set of other mutations, and hopefully we get some insight. But right now for this particular mutation, which is the most famous one, we can already see that it is a key residue to stabilize the drug in the cavity, and the mutation from methionine to valine hinders the binding and results in the drug displacement from the catalytic pocket. So the take-home messages is that these proteins in this family is that they are essential proteins for the biosynthesis, because even though EMBs are already susceptible to ethambutol, these type of studies that I showed, we can get more insight, we can improve the drug maybe, and maybe do specific alterations to the drug or to the compound to make it more efficient. And for AFTs, it's a blank canvas, so we can do drug development on these targets. Acyl carrier protein may be involved in the regulation of all these transferases, and last but not least, docking and molecular dynamics can be powerful tools. We are structural biologists. We like experimental data, but it's very nice to see something on the structure, not a static picture, and if we can trust the technology right now, we can get real insight on drug resistance mechanisms and the impact on the relevant mutations on some TB patients. With that, I would like to acknowledge our lab, my supervisor, Marina Asher. Federico Isobio is helping me a lot with the docking and these simulations. Filippo Mancia's lab in New York for the work with AFTD and the MBB. Tiago Andres with Cytheratic Lab for the SPR assays with the nanobodies. And for the Instruct centers, the Brussels centers of Jan is there in Brussels for the nanobody discovery, and Sarah's Butcher's lab with the hub for AFTA and Impact and all the funding agencies. Thank you.
Okay, now that we finish the session. Now that we finish this section, the presentation part, we now have something called discussion. So I'd like to ask the speakers of the session to come up on stage to answer questions, to ask questions on how they could improve their work, and to discuss with us how to further proceed and to go on. So please join us. You can stay here close to the microphone so that you can answer specific questions that are going to be answered. Thank you. Right, so the discussion session is open, so please ask whatever you want.
So thank you for the nice talks that you presented. So I have the first question for Tiago. So you are the first selected. So I'm curious to know, well, if you know, what is the yield that you get on the cell-free system? So like the amount of protein?
Yes, we know. It's about 2 milligrams of protein per 9 milliliters of medium.
And so, and how long is because I don't know anything about this system, so how long does it take the process itself? So it's except just avoid the part of that you have to submit the Instruct proposal. So like, is it a week time lab or 2 months? So
the preparation of samples, we need to prepare the samples, the plasmids, and send to the Instruct center. And they, for the experiments, they take about 1 month estimated. And then we send again the protein for us. We purify the protein, and it's about maybe 3 months, 4 months.
thank you very much. So I have a second question for Hugo Fraga. Thank you. Just a few participants. So maybe I didn't got from your presentation. Do your protein has so from what I understood, you have like different conformation of different states. Does this correspond to some cellular states, like respond to some stage?
No, there's a mechanism for it's SRUs, not for TB, for TB. There's less studies. There's several protein integration has to be well regulated in the cell. So it's fundamental that you don't degrade what that you don't want to degrade. So one of the hypotheses is that this resting state is a way of preventing also one more step to regulate the degradation. Instead of regulate by having this protease that's linear that is not accessible without the activators, but another step in regulation is to have the activators in this resting state. That's the current. About these two structures that exist in SRs, they have really resolution. 10, and in MTB, we only have these sacks, but this sack stuff is a blob. It's not really something you can. Yeah, to be honest, we put the sacks in the paper because we haven't seen else. We have any more of that's the reality. I'm not gonna lie. It gives us a blob. It's bigger than it should. It fit to the data, so yeah. But it's not my favorite thing.
I have a question for Zé. So I'll ask the same people. Zé, in your last protein, AFTA, you figured out that only about 1% of the particles were the interesting ones.
Yes.
But you showed data for AFTD, where you threw away about 50% of the data. How can you be sure that in those 50% it's not the interesting stuff?
I mean, you never know in the data collection, especially cryo-EM. There you can have conformational states that get separated further down the processing pipeline. So the total particles that you start with, you get very happy when you have a lot of particles because you already know that most of it is trash. That's just the reality of it all, and especially if you get access to data collection, more data collection sessions. Most of the works, for instance, the earlier works on the COVID-19 related stuff, it was millions of particles, and you get like the structure with that 1% of particles from the whole data set. So I guess it's strength by numbers. You can try to improve, and I think there is room for improvement on the sample quality. I think I can shift and increase those numbers for the population that has the complex, but it's never going to be like 50% of the data set. I'm not counting on that. If I get like 15%, maybe I'll do data collection that takes more data, so I can get more particles and increase the numbers by increasing the size of the data set.
If I can have a second one for Sara. Sara, you mentioned resolution for the various imaging methods, but I'd like to know about sensitivity. How many metals have to be very near to each other to actually get a spot on those imaging of X-ray?
I have no idea. I can tell you that comparatively, DDS seems to be less sensitive than X-ray spectroscopy, but I'm not sure you can quantify in terms of numbers of atoms of metal. I'm not sure it's quantitative. I don't think so.
Just curious if having 10 would be enough to get a spot, or if you need to have 100 or 1,000. Just have a rough measure of the range that actually starts to become visible on the detection.
I don't know how to answer that. For sure, I would say 10 is too low. 100 almost for sure is too low. But I think it depends on the technique and on the thickness of the sample, on the penetration of the rays that you are using, so which is related to the thickness of the sample. Yeah, this is my answer. I cannot give any more detail.
Thank you.
Yes. My question is to Tiago also. I would like just if you could explore a little bit more how did you evaluate the interaction between the tetraplex and the nucleolide by fluorescent. And if these outcomes are comparable to the ones that you get by ITC.
Yes, we use a fluorescence spectroscopy. We labeled the RNA, and we titrated the protein without labeling in the RNA solution. And when we see the decrease of fluorescence, it's because the protein is interacting with the G-complex, and the fluorescence decreases because the fluorophore is hived by the protein.
Okay, so it's like a quenching effect that you are
Yes, it's a quenching effect.
Okay,
Thank you very much.
No, sorry. I have a two-part question for Ulu. So the first part is, have you looked into cyclic nucleotides, and do they have any regulatory or inhibitory role in your protein? And the second part is that, have you tried non-hydrolyzable nucleotides to stall your complex?
Well, the ATP, I used to use the non-hydrolyzable nucleotides a lot. This is ClipC, ClipC C, group A, are all part of a family of proteins that includes, of course, 19S of the proteasome, and it's called AA+. I used to use non-normalizable DPMS on a static pan as an analog of 19S. For this kind of approach, for example, there are some proteins that, for example, like DPAX is able to hydrolyze the DPMS also. So I didn't try the non-normalizable nucleotides. I decided to go for what's being used in structural biology of these families, just knock out to make this mutant, this double mutant that produces the eliminates the ATP hydrolysis. Yeah, definitely you could try. You can try other type of nucleotides.
And have you looked into the cyclic nucleotides? Do they have any inhibitory or regulatory effect? Okay, we have some more content. You can discuss it later.
Okay, I have two questions, one for José and another for Isabel. For José, we have heard most of the others speaking about cryo-EM, and you as well, but you were the only one who used nanobodies and megabodies. Looking as far as I know, it's one of the most subscribed platforms in Instruct, and is the unique one in Instruct in Brussels. Would you advise any of the other colleagues or any colleagues on the advantages of the nanobodies and megabodies for their use? And then I have a solution.
That's a loaded question. I think I would advise them to go if you think that it fits your project. I had a wonderful time in Brussels.
No, I'm not asking you that.
Yes, I know, but I mean, it's a good selling point. No, for sure. I mean, the advantages for AFTA, so the complex, even with nanobody, so one step below the megabody, it helped stabilize my protein, even when it was in detergent. So right from the start, I've seen some good improvements in as far as the stabilization of the protein was regarded. We hope that it would even help to in the process of concentrating the protein in order maybe to do some crystallization trials, but the yields were very poor. So we had tons of nanobodies to work, but not enough protein to do crystals. But I mean, they do work. We
think it affects the structure of the protein itself by being bound to such a large volume.
I mean, I still have a low resolution. I mean, it's too early to see. I mean, as far as the purification procedures and the characterization experiments that we do perform with AFTA, the nanobody or megabody being there, I mean, it just it's the same behavior overall, and it even increased stabilization, as I've said. As far as the structural integrity of AFTA being bound to interacting with a second partner, I mean, only when I get the structure I can assess that.
And for Isabel João, are you really tempted now to do cryo-EM studies? Well, how did you get any hints for your work on how to proceed further to structural characterizing your proteins?
Yes. Working with the membrane proteins, we know that for some years it was really difficult to get crystals and protein structures. So it was really interesting to see cryo-EM evolve during these last years and seeing so many new data on this type of proteins being resolved so nicely. So for us, I think we are at the point, for instance, these proteins that are presented, the ADY2, we already have the bacterial homolog structure, so it's not that interesting, but in a recent study, we characterized new carboxylic acid transporters from different families that are not yet, don't have their structure determined, so we only have predicted models. So we want to go ahead with this and try to see, but I think that we are at the point where we have to determine how we are going to work with them. If, for instance, I was hearing about these cell-free systems for protein production. I don't know if they are efficient for membrane proteins or not.
You can do that through instructors, we'll try to find out.
So we have to...
You can profit a lot from instructors.
Yeah, I think we are at the point where we have to explore and try to understand what will be better for our system, because at this point we are working with these proteins that are from yeast, and they have these long loops and this big N and C terminal that were affecting in the past for protein production and crystallization were not very easy to get because of that. So we went to the bacterial homologs, but sometimes they don't work the same. They don't have the same specificities, and the mechanisms are a bit different. So it will be really important for us now to decide which approach we should take and try to take the most of this infrastructure.
I hope next year or next meeting you will be one of the speakers with one structure.
Okay. So thank you for this opportunity.
So any further questions? So I actually have not just it's about Hello? You chose cell-free system to produce. But you have post-cultural modifications and so on. So my question is at what point can you use this infrastructure?
We can use some modifications because we can produce a fusion protein with IBD, for example, to make the protein more solid. We cannot introduce post-translation modification. With some experimental. We can play with.
You can try to calculate
Exactly.
if there was
Exactly. We can use insect cells, for example, or cells. It's better if I have post-translation modifications. They are the best examples. Okay, thank you.
So, any further questions in the room? Any subjects for discussion? Okay, so I suggest that we enjoy the fresh air outside just before coffee, and then perhaps there is a chance to go a little bit deeper into these issues. What I would like to say is that this session showed that we help instruct. Nothing of this would be possible. We can only evaluate how important the thing is, if something is, if we put ourselves in the position of what if instructed? So we would be in the dark. So we need this kind of thing. Okay, thank you very much for this.